Alright, hello everyone. Today we're going to be talking about some more sampling techniques and how that relates to species diversity. So we've already been over the sampling methods. Now here is how you can apply them to investigate species diversity in different areas. So to choose the right approach, when we design and sample a population, two decisions must be made: the sampling method and the sampling technique. So the method we've already kind of gone over. It depends on the habitat. So random sampling, that is best for uniform areas. And systematic sampling, that is better for when you need evenly spaced coverage. Stratified sampling, that suits areas with distinct zones or habitat types. So stratified equals when you have distinct zones or habitat. Okay, and the samples are taken proportionally from each zone. Now the technique depends on what you are sampling. Quadrats, so they're these rectangle things that are split up into 4 sections. They are best used for sampling stationary organisms like plants. So therefore stationary. And you also have line transects. So line transects record species at intervals along a tape. So that is, we record species at these different intervals along a tape. Now, belt transects record all organisms within a defined width either side of a line. So we have 2 lines here, and then we record everything or the specific organism in between. It's useful for gradients like a shoreline. Capture, recapture. So we looked at the Lincoln Index. That capture-recapture model is best used for mobile animals, so things that move around. Okay, now there's an important way to minimize the biases for these different sampling techniques. So to think about the sample size and number. So sample size is better if we have a higher number. So more quadrats and larger quadrats reduce the effect of chance variation. So you increase sample size until the results stabilize. Now you can have random number generators as well. So you can use these to assign quadrat coordinates, removing investigator bias and placement. Now you can also have a thing called counting criteria. Now it defines clearly which organisms are counted before data collection begins. For example, only count organisms with more than 50% of the area inside the quadrat. So that is counting criteria. And it's also important to calibrate equipment and to be precise when taking measurements. So that's ensuring that measuring tapes, quadrats, frames, and any other equipment are accurate. And you also have to remember any limitations that occur when sampling and collecting data. Okay, so you'll use this to calculate species diversity. And species diversity, it accounts for both the number of species present, which is species richness, and it also accounts for the relative abundance of each species. For example, an area with 5 species, all in equal numbers, has a greater diversity than an area with 5 species where one dominates. So when reporting diversity, both of these factors must be considered, not just the total number of species recorded. So that's going back to that SDI value that we talked about in a different video. So to summarize, it's important to match the sampling method and technique to the habitat and organism. Minimize bias through sample size random placement, clear counting criteria, and calibrated equipment. And it's important to report diversity in terms of both species richness and the relative abundance of each species. All right.